Guys, we have been talking about pancreas, insulin, and diabetes. Pancreas, the endocrine part of the pancreas, consists of pancreatic islets of Langerhans, which are dispersed throughout the exocrine portion of the pancreas. The islets secrete three hormones: insulin, glycogen, and somatostatin, which helps regulate the blood levels of nutrients, especially glucose. Alpha cells secrete glycogen, beta cells secrete insulin, and delta cells secrete somatostatin. It's very important to maintain blood glucose level within a normal range. A below normal blood glucose level causes the nervous system to malfunction because glucose in the nervous system's main source of energy. You know it. When blood glucose decreases, other tissues rapidly break down lipids and proteins to provide an alternative energy source. As lipids are broken down, the liver converts some of the fatty acids to acidic ketones, which are released into the blood. When blood glucose levels are very low, the breakdown of lipids can cause the release of enough fatty acids and ketones to reduce the pH of the body fluids below normal, a condition called acidosis. The amino acids of proteins are broken and used by the liver to synthesize glucose. The hormone insulin is released from the beta cells primarily in response to the elevated blood glucose levels and increased parasympathetic stimulation associated with digestion of a meal. Increased blood levels of certain amino acids also stimulate insulin secretion. A decreased insulin secretion results from the decreasing blood glucose levels and from stimulation of the pancreas by the sympathetic division of the nervous system, which occurs during physical activity. Decreased insulin levels allow blood glucose to be conserved to provide the brain with adequate glucose and to allow other tissues to metabolize fatty acids and glycogen stored in the cells. The major target tissues for insulin are the liver, adipose tissue, muscle, and the area of the hypothalamus that controls appetite, called the satiety center. Insulin binds to membrane-bound receptors and either directly or indirectly increases the rate of glucose and amino acids uptake in these tissues. Glucose is converted to glycogen or lipids, and the amino acids are used to synthesize protein. Diabetes mellitus has several causes. Type 1 diabetes mellitus occurs when too little insulin is secreted from the pancreas, and type 2 diabetes mellitus is caused by insufficient numbers of insulin receptors on target cells or by defective receptors that do not respond normally to insulin. In type 1 diabetes mellitus, tissues cannot take up glucose effectively, causing blood glucose levels to become very high, a condition called hypoglycemia. Because glucose cannot enter the cells of the satiety center in the brain without insulin, the satiety center responds as if there were very little blood glucose, resulting in an exaggerated appetite. The excess glucose in the blood is excreted in the urine, making the urine volume much greater than normal. Because of excessive urine production, the person has a tendency to become dehydrated and thirsty. Even though blood glucose levels are high, lipids and proteins are broken down to provide an energy source for metabolism, resulting in the wasting away of body tissues, acidosis and ketosis. People with this condition also exhibit a lack of energy. Insulin must be injected regularly to adequately control blood glucose levels. When too much insulin is present, as occurs when a diabetic is injected with too much insulin or has not eaten after an insulin injection, blood glucose levels become very low. The brain, which depends primarily on glucose for an energy source, malfunctions. This condition, called insulin shock, can cause disorientation and convulsion and may result in loss of consciousness. Fortunately, genetic engineering has allowed synthetic insulin to become widely available to diabetics. Glycogen. Glycogen is released from the alpha cell when blood glucose levels are low. Glycogen binds to membrane-bound receptors primarily in the liver, causing the glycogen stored in the liver to be converted to glucose. The glucose is then released into the blood to increase blood glucose levels. After a meal, when blood glucose levels are elevated, glucagon secretion is reduced. Our next hormone is somatostatin, is released by the delta cells in response to food intake. Somatostatin inhibits the secretion of insulin and glucagon and inhibits gastric tract activity. Insulin and glucagon together regulate blood glucose levels. When blood glucose levels increase, insulin secretion increases and glucagon secretion decreases. When blood glucose levels decrease, the rate of insulin secretion declines and the rate of glucagon secretion increases. Other hormones such as epinephrine, cortisol, and growth hormone also maintain blood levels of nutrients. When blood glucose levels decrease, these hormones are secreted at a greater rate. Epinephrine and cortisol cause the breakdown of protein and lipids and the synthesis of glucose to help increase blood levels of nutrients. Growth hormone slows proteins breakdown and favors lipid breakdown. So guys, now we're gonna be talking about testes and ovaries and their hormones. The testes of the male and the ovaries of the female secrete sex hormones, in addition to producing sperm cells or oocytes respectively. The hormones produced by these organs play important roles in the development of sexual characteristics. Structural and functional differences between males and females, as well as the ability to reproduce, depend on the sex hormones. The main sex hormone in the male is testosterone, which is secreted by the testes. It's responsible for the growth and development of the male reproductive structures, muscle enlargement, the growth of body hair, voice changes, and the male sexual drive. In the female, two main classes of sex hormones secreted by the ovaries affect sexual characteristics: estrogen and progesterone. Together, these hormones contribute to the development and function of female reproductive structures and other female sexual characteristics. Two such characteristics are enlargement of the breasts and the distribution of adipose tissue, which influences the shape of the hips, breasts, and the thigh. In addition, the female menstrual cycle is controlled by the cyclical release of estrogen and progesterone from the ovaries. Luteinizing hormone LH and follicular stimulating hormone FSH stimulate the secretion of hormones from the ovaries and testes. Releasing hormone from the hypothalamus controls the rate of LH and FSH secretion in males and females. LH and FSH in turn control the secretion of hormones from the ovaries and testes. Hormones secreted by the ovaries and testes also have a negative feedback effect on the hypothalamus and anterior pituitary. Alright guys, subscribe to my channel please, give your comments on comment section, give me a likes, bye bye, see you.